This is me Arju here and I welcome you to my channel Mr. Teeth, where your dental concept get dissected. So this is our part 2 video of the development of the teeth, and in this video we will be discussing with the morphological stages of the tooth development, and in this topic we will be discussing particularly with the bud stage and the cap stage. Okay, so before starting today's video, I will be doing a small recap of yesterday's video, then I will start with this today's topic. Okay, so as I have told in my previous video there, the primary epithelial band was formed there. Okay, so after the formation it was formed in ectomesenchyme. Okay, ectomesenchyme is the connective tissue that is just beneath the stratified squamous epithelial layer. Okay, that is present in a stomodium, and below that in the ectomesenchymal connective tissue, okay, there is a formation of the primary epithelial band, and from primary epithelial band there develops the lingual process and the buccal process, and the lingual process there is a formation of the dental lamina, and the buccal process there is a formation of the vestibular lamina. Vestibular lamina forms the vestibule, and the dental lamina, okay, it forms the deciduous teeth, okay, with the 3 permanent posterior molars. Okay, and after that, when this dental lamina gives the lingual extension, that is forming the successional lamina, and that successional lamina it gives the development. Okay, it gives the development of the permanent dentition. Okay, so we have discussed this much in our first videos of the development of the teeth. So in today's videos we will be discussing with the bud stage and the cap stage. So without delaying much, let's get started. So in this first line, okay, as I have mentioned here, the morphological stages of the tooth development. Okay, so simply morphological stages of the tooth development means we classify the stages. Okay, we divide the periods of the tooth development in different periods. Okay, we divide the development of the tooth in different periods, okay, based on their morphology. Okay, morphology means shape and size. Okay, on the basis of the shape and size of the enamel organ, we just classify or divide the period of the tooth development, and they are properly 3 stages. Okay, so first stage is our bud stage, and second stage is our cap stage, and third stage is our bell stage. And in bell stage we have early bell stage and advanced bell stage. Okay, so in this video we will be particularly focused on bud stage and cap stage. In later video we will be discussing in detail about this bell stage. Okay, so in bud stage, as I have written here, the bud stage, it is a epithelium of the dental lamina is separated from the underlying ectomesenchyme by a basement membrane. Okay, as I have discussed earlierly in my first video. Okay, so there is a stratified squamous layer. Okay, so there is a stratified squamous layer present. Okay, there is a stratified squamous layer present, and beneath that there is a ectomesenchyme. Okay, so the epithelium of the dental lamina is separated from the underlying ectomesenchyme by the basement membrane. So from this there form the primary epithelial band and dental lamina. So dental lamina was like this. Okay, if this is a epithelium, then there is a like this structure was there. Okay, so I am drawing a schematic diagram. Okay, so this structure is a dental lamina. Okay, and this dental lamina, this hollow structure. Okay, so there is a lining present there. Okay, and that lining is separated from the ectomesenchyme, okay, by the basement membrane. So this is a basement membrane separating the epithelial lining of the dental lamina and the ectomesenchyme. And after that, in that basement membrane, okay, so in that dental lamina there is a round to ovate swelling arise from it at around the 10 different places. Okay, so from that basement membrane, okay, there arise the round to ovate swellings, okay, at 10 different places. So it means that 10 different, why 10 different places? Then okay, in primary dentition we have 20 tooth. Okay, so we have 20 tooth, and 20 tooth means 10 plus 10, so 10 in maxillary arch and 10 in mandibular arch. So there is swelling in the 10 different places. Okay, and that swellings are called enamel organ. Okay, so previously also in my first video I have just discussed little bit about the enamel organ, but here just you understand that. Okay, so there is a dental lamina, and in dental lamina, okay, there is a epithelial layer, and that is separated from the beneath ectomesenchymal connective tissue by the basement membrane, and from that basement membrane there arises a 10 different places there arises a round or ovate swelling, and that round or ovate swellings are called the enamel organ, and they are responsible for the deciduous dentition. Okay, for formation of the deciduous teeth. Okay, So after this, okay, we will be discussing with this point. In this point, as I have written here, shape of the enamel organ is bird shape, so this stage is called body stage. Okay, so this, as I have told here, okay, so on the basis of morphology, we are just dividing the periods of two development. Okay, so in body stage, the morphology of the enamel organ is of that of the bird. Okay, so there is a morphology of the bud. Okay, so bud means as there is a flower bud. Okay, you have seen or not. Okay, so the enamel organ that is present in this stage is just similar to that of the flower bud. After that, I have written here the enamel organ contain two different types of the cells. So there are two different types of the cells are, one is a peripheral cell and another is a central cell. So this peripheral cell means the cells that are in the periphery. Okay, and the central cells means those cells that are present in the center. Okay, so this enamel organ, okay, so I will show you by the another pen. So in this enamel organ, the cells that is here, okay, so this green structure, green line that is present here, so it is a periphery of the enamel organ. So the cell that is present at this peripheral portion is the peripheral cell, and they are low columnar to cuboidal. Okay, so this is very very important, you should understand this and also remember this. Okay, they ask you in viva, so what is the shape of the cell, okay, that is present in the periphery of the enamel organ in the body stage, then you should tell cuboidal or the low columnar. Okay, so it is very important. Similarly, the central cells, okay, so the central cells mean, so, okay, I will be showing by the red pen. So central cell means the cell that is present in between the enamel organ, okay, in the center of the enamel organ, that is surrounded by this green color cells. Okay, so the green color is peripheral cells and the red color is a central cells, which is a polyhedral in shape. Okay, polyhedral or polygonal in shape. Okay, so we have completed this enamel organ cells. After that, I have written here the condensation of the cells that is adjacent to the enamel organ. Okay, it is due to the, which is due to the increased mitosis and migration of the neural crest cells to the adjacent areas of the enamel organ. Okay, so this is very important to understand here. Okay, so if this is an enamel organ, okay, so the cells that are around this, okay, so the cells that are around this, I will show you by a picture that I have shown you in previous video. Okay, so there I have drawn a proper picture. Okay, so in this picture, as you can see here, okay, so this will be easy to understand you. Okay, so this structure is an enamel organ. Okay, and this, okay, so this peripheral cell in this body stage, okay, is the cuboidal or the low columnar, but as you can see here, it is columnar here and cuboidal here. Okay, so I will be discussing with this in a cap stage. Okay, so don't get confused, this picture is of cap stage. Okay, but I am discussing with the body stage now, so that you should be relating that. Okay, so this enamel organ is bird shape. Now just think like that, and there is a cell that is a low columnar cuboidal in a periphery and polyhedral in central. So I was discussing with this point. So in this point, as I have written here, it is due to the increased mitosis and the migration of the neural crest cell to the adjacent areas of the enamel organ. Okay, so as you can see, the cells that are around this enamel organ are condensed. Okay, so they are condensed connective tissue. Okay, so those connective tissue cells, okay, that are present here is due to the increased mitosis. Okay, so there is this connective tissue cells that are present in this connective tissue ground substance, so it increases its mitosis, it proliferates in high manner, okay, in fast manner, and it do condensation. Similarly, there is also the migration of the neural crest cells. Okay, so the other connective tissue cells, neural crest cell, ectomesenchymal cells, okay, it migrate from other side to here. Okay, and adjacent areas of the enamel organ, so there is a formation of this condensation of the connective tissue around the enamel organ. So they both are the reasons behind the condensation. Okay, so you should remember this. After this, in our next paper, so as I have written here, so condensation, there are two type of condensation in this body stage. One is ectomesenchymal condensation, that is adjacent to the enamel organ or in the invagination of the enamel organ. Okay, so invagination of the enamel organ means if this is a enamel organ, okay, so this is an invagination present here, and the condensation that is present here. Okay, so this condensation is the ectomesenchymal condensation in the invagination of the enamel organ, and that structure, okay, so that condensation is called the dental papillae. Okay, similarly, the marginal condensation of the ectomesenchymal cell enclosing the dental papillae and the enamel organ, okay, that is called dental follicle or the dental sac. Okay, so we have completed this. After that, so you should remember this, what is dental papillae and what is dental follicle. Okay, some may think that, okay, so dental papillae, okay, some may things like that. Okay, so dental papillae is the cell that is just beneath this invagination, and the dental follicle are the cells that are outer to the dental follicle, but is not so. Okay, it is so, but it also include the cell that is around the enamel organ too. Okay, so it is just cells that is encircling both the structure enamel organ and the dental papillae. Okay, and this all structure is called tooth gum, as I have discussed in my previous video. Okay, now I have written here about the morpho differentiation and the histo differentiation. Okay, so morpho differentiation means just think like that. Okay, so if any structure is changing its shape, okay, so just there is changing in a shape, then it is called morpho differentiation. Change in shape of the enamel organ is there, then it is called morpho differentiation, which is present in this body stage. But histo differentiation means the change in the shape. It is not change in the shape, sorry. Okay, it is the specialization of the enamel organ. It means that, okay, so the cells of the enamel organ would differentiate into certain other cells, okay, which have some particular functions. Okay, so that is called histo differentiation. So just get uh, don't get confused in those points. Okay, so morpho differentiation is present in the body stage, but histo differentiation is absent in the body stage. So we have completed with the body stage. Now I will be discussing with the cap stage. Okay, so in the cap stage, as I have written here, so cells of the enamel organ proliferate but doesn't expand uniformly into the larger sphere. Okay, so but unequal growth in the different parts lead to the cap formation and gives the illusion or the appearance of the enamel organ wearing a cap. Okay, so as this is a, okay, so I will be showing here only. So if this is a, okay, so if this is a epithelial layer, okay, so if this is a epithelial layer and this is the enamel organ, okay, and this is a ectomesenchymal condensation surrounding this enamel organ. Okay, so what happens here is that, okay, previously in our body stage, okay, previously in a body stage, it was just like a bird. Okay, it was just like a bird. I am doing just a schematic diagram. Okay, so okay, this is a ectomesenchymal condensation here. Okay, this is ectomesenchymal condensation here. Okay, and this is a bird like structure. So this is a bird stage and this is a cap stage. So you can see here change in the shape. Okay, it is due to the unequal growth. Okay, if there is growth of this body stage, then there would be larger sphere. Okay, right, but there is not a larger sphere, there is just a structure that is similar with that of the cap. Okay, it means that some structure are growing in higher pace, but some structure are growing in the lower pace. Okay, so that it forms the cap stage. There is unequal growth forming the cap. Okay, so cap formation is there. Okay, and that is giving the illusion of that enamel organ. So this dental follicle is wearing the dental follicle and the dental papillae are wearing, are acting as a cap that is okay, worn on this enamel organ. Okay, so we call cap stage. After that, okay, the cells, okay, that are present in this cap stage are, that is outer and inner enamel epithelial cell. Okay, so as in the body stage there was no any such cells like this outer enamel epithelium, OEE. Okay, so we also call this OEE or IEE. Okay, so there was no any such cells, there was just a peripheral cells and the central cell. But here you can see here there is a outer enamel epithelial cell and inner enamel epithelial cell. So outer enamel epithelial cell or the peripheral cells are cuboidal here, and that covers the convexity of the cap. Okay, so this is a, I'll show you by the another pen. So okay, so this is a convexity. Okay, so this is a convexity. So this convexity of the cap stage, okay, sorry, enamel organ is having the cuboidal type of cells. Okay, which is in the periphery, right? Okay, so it is outer cell, outer enamel epithelial cell that is in a periphery of cuboidal shape covering the convexity of the cap. Similarly the inner cell, that is columnar. Okay, so I will show you by the another pen. Okay, so the inner cells, okay, that is columnar. Okay, so this is a inner cell and that is columnar, and that is taller cells that is present in the concavity of the cap. Okay, So it is in a concavity. So these are inner enamel epithelium and these are outer enamel epithelium, and this outer enamel epithelium is cuboidal in shape. Okay, so this is cuboidal in shape, and this inner enamel epithelium is columnar or taller cells. Okay, now as I have written here, both the cells are separated from the connective tissue by the basement membrane. Okay, so it is easy to understand. So this red line is acting like a basement membrane that is separating it from the surrounding connective tissue. Okay, so we have completed this point. Now in another paper, as I have written here about the stellate reticulum. Okay, so there is a special cell that is called stellate reticulum. Okay, that is star like cell. Okay, so it is star like cells. Okay, so stellate reticulum or the star like cell is a distinguishing cell. Okay, that appears in the cap stage of the tooth development. So stellate reticulum, how it develops is that, so the polygonal cells that are located in the center of the epithelial enamel organ, also called enamel pulp. Okay, so stellate reticulum is also called the enamel pulp. You can remember this enamel pulp. Okay, so enamel organ pulp or enamel pulp you can say. Okay, so it is star shaped cells. Okay, so how it is formed is that, okay, so the polygonal cells. Okay, so the polygonal cells, as I have discussed in a bird stage, there was a polygonal cells in the center of the enamel organ. So those polygonal cells of the enamel organ, okay, it releases the glycosamine glycans. Okay, so glycosamine glycans, okay, or the GAGs you can see. So glycosamine glycans are released in the intercellular areas, and those intercellular areas, when it have this GAGs, okay, so they are hydrophilic also. Okay, so just think. Okay, so these GAGs are released in intercellular areas, and those GAGs are hydrophilic. Okay, so when they are hydrophilic, it is of sure that it will absorb the water. Okay, so it absorbs the osmotic force and absorbs the water from the surrounding connective tissue, and that water would cause the accumulation of water in the intercellular area. So there will be increase in the content of the water in the intercellular area that would push the cells, okay, apart but remain attached by the intercellular bridges. Okay, and there is appearance of the star like cell. Okay, so the thing that I wanted to explain here, and this is very important to understand. Okay, so just think, this is a polygonal like cells. Okay, just I am drawing two pictures, two cells. Okay, so this is a polygonal cell. Okay, so now what happens is that it releases the GAGs. Okay, so GAGs is released in intercellular space. So here is GAG released, and that GAG is hydrophilic, and that hydrophilic GAG would absorb the water and would compress those cells. Okay, so compress those cells. Okay, so it will be like this now. Okay, so okay, sorry, so it will be like this. Okay, so it is connecting here. Okay, so it is compressed here. Okay, and being connected at this junction, that is intercellular bridge. Okay, it is connected at the intercellular bridge. Okay, so it is appearing like the star shape. Okay, but it was polygonal earlier. Okay, so it is due to the glycosamine released from those polygonal cell. So those types of cells that are star like, okay, that is called the stellate reticulum cells. Okay, star like cells. Now in another point, as I have written here, due to the collection of the water, it gives the cushion like consistency, which delicates, okay, which provides the protection of the delicate inner enamel forming cells. Okay, and it acts as a shock absorber. Okay, so it is easy to understand. So it acts as a cushion. Okay, it provides the protection to the enamel organ cells. Now in another point, okay, so this is also very important to understand here. Okay, so in the enamel needs. Okay, so the enamel needs means, okay, so enamel needs or enamel needs, what you pronounce. Okay, so enamel organ is seen to have double attachment of the dental lamina containing the entrapped ectomesenchyme, and appearance is due to the funnel shape depression, curving depression or the curving of the dental lamina. Okay, so I will just explain this terminology, which is very important, in very simple term. Okay, so what happens is that, okay, so in this picture I have drawn here. Okay, just think this is a dental lamina. Okay, and this dental lamina is connecting the oral epithelium with the enamel organ, and this is a curved structure. Okay, so this is a curved structure. So when we will cut this, okay, so when we cut this, and when we will see it in the microscope, we can see that there are two sides. Okay, so it is having the two sides, but we know that it is just a one structure. It is a structure that is in a straight line. Okay, sorry, not in a straight line, but it is a structure that is just a one structure. There is not two dental lamina present in the enamel organ, but when you will cut this, okay, you can see the two dental lamina are present there, okay, two strips, okay, two strikes of the dental lamina, okay, is present there, okay. So you may confuse that either there is presence of the two dental lamina, but it is not so, okay. So this is very important here. So enamel niche is an artifact like, okay, structure, okay, that we can see in the microscope, and what we see is that, okay, so it is a double attachment, okay. So we see the double attachment of the dental lamina. We can see that there are two dental lamina that are just connecting the epithelium and the enamel organ, but it is not so. So it contains the, so in this space, okay, so in this space it contains the mesenchyme. So it contains the entrapped ectomesenchyme. In appearance is due to the funnel shape depression or the curving of the dental lamina. So this is a, okay, so there is curving, and when we cut, so you can see two dental lamina present there. So that is called enamel niche, okay. So I hope you got cleared in this concept. Now I will move forward. So now we will be studying the temporary and the transient structures of the enamel origin, okay. So it is formed in a cap stage and disappears after the enamel formation begins, okay. So when there is formations of the enamel, when there is a mineralization, okay, that is in an advanced belly stage, okay. So at that time it is just lost. So the structures, so the temporary and transient structures are the enamel knot, enamel cord, enamel septum, and enamel label, okay. So those transient structures means those structures that are present just for a small time period, okay. They are not that are not present for the larger time period. So they are just present for the small time frame, so they are called transient structure. So our first structure is the enamel knot, okay. So the enamel knot, there is a dense collection of the cells at the center of the enamel organ, okay. And when that enamel knot, okay, is extending vertically towards the another side of the outer enamel, I will show you by the picture, drawing the picture, okay. So just think that, okay, so I will show you the picture, okay, so then it will be easy. Just think, this is a, okay, so this is the epithelium layer and this is a dental lamina, and just think this is a enamel organ, okay. Just I am drawing just schematic diagram, okay. So don't you get confused that this is sphere and we are discussing with the cap stage, okay. So it is just a matter of understanding. So this is a enamel organ. So now what happens is that, so enamel knot is a transient structure, so there develops the extension, okay. So there develops the extension, okay. So this is an extension that is developing inside the enamel organ, and that is the enamel knot. There is a dense collection, okay. So at this side there is a dense collection of the cell, and when this enamel knot, okay, is extending and growing, okay, is extending and growing towards the outer enamel epithelium. So this is outer enamel and this is the inner enamel, okay. Just think like that, okay. So this is growing towards the another side of the outer enamel epithelium, okay, and that extension, that vertical extension is called enamel cord. And after that, when this enamel cord would reach the outer enamel epithelium and just form the two hollow spaces, okay, so it forms the two compartments in the enamel organ. At that time it is called enamel septum, okay. It extension of the cell reaches the outer enamel epithelium and divides the stellate reticulum into the two parts, okay. So and after that, okay, so where this enamel cord, okay, it touches the outer enamel epithelium, at that side there is just a like this, okay. If this is a, okay, so like this structure is formed there, okay. So it means that outer enamel epithelium is also moving towards the enamel cord side, okay. So there is just a concavity formed, okay. There is a concavity formed at the outer enamel epithelium where the enamel septum makes the contact, okay. So concavity is formed near the outer enamel epithelium. So just think, enamel knot means it is a dense collection of the structure at the center of the enamel organ, okay. So there is a dense collection of center of the enamel organ. After that it moves towards the outer enamel epithelium and it is forming the septum later. And after when the septum is formed, okay, so there is formation of the two compartment, and at the same side when the enamel septum touches the outer enamel epithelium, there is a concavity developed in outer enamel epithelium, that is called enamel navel, okay. So just remember all those transient structures. Is it okay? So the functions of this enamel transient structure, okay, enamel cord, enamel knot, enamel septum is that, okay. So they just act like a reservoir, okay. They may act as a reservoir of the dividing cell for the growing enamel organ, okay. So they just act as a reservoir of the dividing cells. After that, recent studies have also shown that enamel knot, okay, so that act as a signaling center as it many important growth factors are expressed by the cells of the enamel node, and they play important part in the determining the shape of the tooth. Okay, so we can remember just two functions. So the particularly the transient structures and the temporary structures, okay, like enamel node and enamel cord. Okay, so they have particularly the reservoir function. So just they reservoir the cells. Okay, so they just store the cells, dividing cells in the growing enamel organ. But when those enamel node are developed in okay, so in when there is a proper development of the enamel node or the cord, they act as a signaling center and they release the growth factor, and those growth factors are expressed by the enamel node, and they help in the determining the shape. Okay, so those growth factor would act as an important thing that is causing the determination of the shape of the tooth. Okay, just remember like that. After that, I have written here about the morpho differentiation and the histo differentiation. So in the previous stage, that is in a bud stage, there was just a morpho differentiation but no histo differentiation present there, that is no specialization of the cell was there. But here you can see the both the morpho differentiation and the histo differentiation is present here. Okay, so just remember with this point, they can ask you in the viva. It is a very important question to be asked in viva. Okay, so in this dental papillae, okay, so under the organizing influence of the proliferating epithelium, there is ectomesenchyme further proliferates and connects, and there is active budding of the capillaries in this dental papillae, and the mitotic figures are also seen there. After that, the peripheral cells adjacent to the inner enamel epithelium, it enlarges and later differentiates into the odontoblast. Okay, and in this dental follicle or in the dental sac, okay, so it shows the further condensation of the ectomesenchymal cells. Okay, and it becomes more fibrous and denser in the cap stage. Okay, so this is about the dental papillae and the dental follicle or the dental sac in the cap stage. Okay, so as we have covered all those points, okay, in this cap stage and the pod stage that are to be discussed in this video. So for today, this much, and we will meet in the next video, next part of this development of the tooth. Till then, stay happy, stay healthy, stay tuned. Okay, so I will be uploading the part 3 of this tooth development very soon. Also, the thing that I missed to say, if you love this video, please do like, share and subscribe. Okay, so just share with this video with your batchmates so that they could also see this video. Okay, so tata bye bye, thank you.